Thank you, everyone. Thanks for coming back on time after the exciting morning session and our lunch. And so with this audience and our audience online, we don't need to go over all the reasons as to why data management and sharing. For this panel, we have prepared together and defined the scope to be the entire life cycle of data, and the goal to achieve by having this panel discussion is to what can we do, how can we do the best in the near future and down the road to enable in-depth scientific discoveries that were able to be made previously before the big data era, and discoveries that can only be made with multidimensional and multi-model data. So that's the scope and the goal for this panel. And let's do a quick introduction, and then we'll dive into the questions to be addressed by our experts that will benefit us with their experiences and their insight. I'll introduce myself. Again, my name is Wang. Hello, everyone participating remotely. I am the program director of the FaceBase program that is now co-founded by NIDCR, NIDCD, and the NIHODSS office. I'm standing here up the stage representing the whole team of the NIDCR program staff, and also always appreciate the guidance from our Institute Director, Jennifer. My background is in transcriptional regulation research with a focus on the immune system development and genomics. And at NIDCR, I have been developing translational genomics and data science. And the next is
thank you. Hi everyone, my name is Vidhya. I'm NIDCR's Chief Data Scientist. My background is that I'm a dentist, a radiologist. I have a master's in epidemiology, and then my PhD is in bioinformatics in the genetic epi of oral cancer and factors. I did my thesis primarily using the UCLA Atlas Biobank, which is a biobank based off of electronic health records and genetic data. So my expertise is in both of those areas. And at this, at NIDCR, I try to put all of that together to help programs like FaceBase, and I work very closely with our senior advisor of data science and our acting director to support NIDCR's mission.
Thank you, Vidhya. Anup, your turn.
My name is Anup Mahurkar, and I wear two hats. One of my jobs is which is my primary appointment, where there's an Institute for Genome Sciences where we do DNA sequencing. And my role there is to develop visualization tools in the space of genomics and some clinical data as well. And with NIDCD, I serve as an advisor to the director on data science and data sharing.
Thank you. Troy.
Okay, my name is Troy Hackett. I also wear two hats at NIH and NIDCD. I work with a team of program officers on three projects that are part of what we call the Temporal Bone Initiative. So these projects, which includes ear-base, are designed to facilitate research in hearing and balance disorders. My laboratory at Vanderbilt University is focused on transcriptomics, the impact of aging and noise exposure on different parts of the auditory pathway.
Thank you, Troy. Axel.
Hi, my name is Axel Visel. I'm at Lawrence Berkeley National Lab. Our lab works in functional genomics in the mouse system. I will not talk about that much more because Laura will give an overview of our work in the afternoon session today. I'm here for two reasons, I guess. So I've been part of FaceBase Program lab has been a major data producer. Later on, now involved as an advisor. But I also served as the chair of the NIDCR Data Science Strategy Working Group for the last few years, which has looked in depth at the data science strategy of NIDCR. And I will also be making some comments here from my perspective.
Thank you, Axel. Mike?
My name is Mike Feolo. I'm a scientist for NIDCR. I previously worked at NCBI for 25 years. I was the team lead for the dbGaP repository. And my background is in genetic epidemiology and bioinformatics.
Thank you, Mike. Carl?
Let's see, I'm Carl Kesselman. I'm the Keck Professor of Engineering at the University of Southern California and the Director of the Informatics Systems Research Division Sciences Institute. I'm the co-PI of FaceBase along with Yang, and I've had the pleasure of dealing with this group for now 12, 14 years.
Yeah.
And my job is to stay out of Rob and Andro's way.
Thank you. So everyone on the panel, please keep in mind the scope and the ultimate question that we raised for our panel. My first question will go to Troy. As a seasoned investigator, what would you consider to be a optimal data repository to enable you to carry out your research the best possible way?
Okay. Well, ups and downs. First of all, a couple disclaimers. I'm not a data engineer, computer scientist, anything like that. I'm not a database aficionado. It's not my hobby to do databasing, but I'm someone who, in my own research and now with your base, is deeply interested in databases that are useful. And so for me, in my own work, there's two databases that I've used the most in my career. One is the Allen Brain Atlas. You may not think of that as a database, but I do. The other one is GEAR, which is at the University of Maryland, which is co-founded and directed by Anup here. That focuses on transcriptomics and auditory systems. For me, what is an ideal database? Well, GEAR and Allen Brain, for me, have been examples of the idea. And that is, number one, they contain data. It seems like a joke, but there are so many databases out there that I have searched and tried to use that don't contain very many useful data. And so at the end of the story, if a database may exist, it may be on the web, you may be able to find some things, but many of them are not updated regularly, or they're not used very widely, and so they don't have much utility. So that's number one. Number two, the data that are on a database need to be related in some way and in a meaningful way. So the way I think about it is this. So if there are maybe 1,000 data sets on some database, but they're minimally related to one another or only related in one feature, let's say the methodology in which the data were collected, very often that means that there are 1,000 little data repositories on that site, and the data are not related to one another in a meaningful way. So that's what I like about Allen Brain and GEAR, and then I've been able to sit in front of those and with those sites for hours, dive deep, or just browse casually, and the data are so internally linked to one another that I can come out of that deep dive with a lot of information. So those are the things that I find most.
Thank you. So, is our satisfying GEAR user, Anup, tell us, how did you reach this level of success? What specific lessons learned in a positive way that you could share with us?
Sure. So I'm going to take a little bit of a detour to give a little bit of history of GEAR, because this is a new audience who may not be familiar with GEAR. So GARE was essentially co-founded by surgeon scientist, her name is Dr. Rona Hartsano. And she at that time was at University of Maryland in Baltimore, where she had a basic science lab as well as she was an operating surgeon. And she came to me because she was generating transcriptomics data. And as a clinician, she had no way of using bioinformatics tools to visualize the data that her lab was generating. And this is about 14 years ago. And she said, Anup, you're an engineer. Can you build me a tool where I can compare my gene expression data to some other lab's gene expression data to sort of figure out what, how do this data compare? What is with one particular gene I'm interested in? How does it behave in other data sets? And so out of that grew GARE. And so we've been building GARE for close to about 10 years now, 10 to 12 years now. And it essentially has about 1,200 data sets. And our data sets that are very small. And when we mean data sets, we mean more one experiment that has been run, maybe in a mouse model or a zebrafish model or a human model. And so we bring that in, and we have about 1,200 of those data sets. It's about 2,000 users who sort of periodically come in, sort of browse the way data is being browsed by Troy. And so the main approach we took there was to make it usable to users who are not informaticians. We try to present side-by-side gene expression across experiments. So you could essentially look at what was happening in microarrays. You could look at it, what's happening in a single cell, as well as bulk RNA-seq. But you can essentially visualize the gene expression. Another innovation was we actually added a cartoon shape so that you can actually think about the spatial transcriptomics now, right? People want to see where in geography in a tissue is gene expressed. So we took a very different approach 10 years ago where we said, okay, create a cartoon of the cochlea. And based on, people were using FACS then to do cell sorting. So they would sort of visualize where is a single cell in the cochlea spatially and what was its gene expression. And so that's kind of what made GARE very different than some of the other gene browsers, because there have been gene expression browsers, there are lots of them around. But that was a different approach that GARE took, which is to make it easy for a biologist who doesn't know computer program to come and look at gene expression across the data sets. So that's sort of the background in the detour. Now, to address a specific question about what is successful, I'd say there are 2 or 3 things that make GARE very successful. One is we know our audience. We are trying to not cater necessarily to an informatician. It's the biologist. So what can we do for the biologists to make their lives easier? The second approach we took is you need an advocate. So Rona Hartsano, being a surgeon scientist, was at meetings talking about GARE, networking with people, trying to sort of solicit people to submit their data sets into GARE, because that's the big success is how do you sort of encourage people to use that platform? And it becomes a network effect, right? If you just have 1 or 2 data sets, it's not going to create a network. But with 1,000 data sets, then people are going to come back to it again and again. So how do you create a network effect for a repository? We call ourselves as a knowledge base because we actually are not a repository in the traditional sense. So for the knowledge base, we try to sort of, whenever a paper is published in a particular sphere, we reach out to the PIs and try to get their data into GARE. That's how we did in the beginning. And now actually NIDCD encourages everybody to deposit the data into GARE. So because of that, we've actually become very successful in creating all these data sets. And we have, I think last count was something like 60 or 70,000 different pages every year from these 2,000 users. And everybody spends somewhere around 12 to 15 minutes on some of our papers. It's a very well-used resource, but those are some of the things which led to our success.
To me, that's a very informative sharing of your success, and it does speak to what Troy would consider the ideal data repository.
Indeed.
Going forward, if I could just add a bit, going forward, when gene expression from the gene RNA to protein and down the road, the effect at the level of metabolics, etc. What to consider the major challenge for GARE to evolve with your data users that GARE has been targeting specifically?
Now, that's a great question. So in the beginning, GARE was very gene expression focused.
Right.
And over the last, I would say, 5 years, we have essentially tried to bring in epigenomics data. So more upstream to try to see what's happening at the methylation level or the open chromatin level. But what we are trying to figure out, but next 5 years, we are going to start delving into proteomics and metabolomics. One of the challenges we have discovered is looking at a gene is very easy, but trying to see what protein is created from that gene becomes much more complicated, right? It's not a one-to-one relationship. So how do you kind of visualize the cause and effect from gene to protein to a metabolite? And how do you navigate that? And that's kind of where we're trying to sort of see what can we innovate to make it easier for a user to make that journey going from all the way from your, I wouldn't say gene level at the genome level, but definitely at the epigenome level to transcriptome, to proteome, to metabolite. That journey is sort of the part.
Thank you. That makes a lot of sense to me. Oh, your time is up.
Just in time.
Okay, so not that I don't want to give our audience any opportunity to ask or to discuss with our panelists. In the interest of time, let's just give each one of them the opportunity to share first. And then if there's time, we could, support a couple of questions, or there's dinner to keep discussing. So the next question goes to Mike, our data repository expert. And what is your lessons learned to share with us? And if you were charged to stand up dbGaP now, what would you have done different?
Okay, so I wrote down some notes, thinking that you might ask me something similar to this. So I would say there's kind of three things that we did pretty well in dbGaP, not perfect, obviously. And the first one had to do with creating a very generalized submission model so that we could go to researchers regardless of the data or the study that was funded. That model would be adequate to take in data. This is, the time back in 2007, this is like when the iPhone came out. To give you some perspective, there wasn't a lot of other resources around that we could compare to. There were a number at NCBI, and we had the infrastructure set up at NCBI. I'm no longer at NCBI, but I would have to say that the resources available to us were limited. And I'll just leave it at that. But that generic submission model allowed us to connect with a lot of different research groups. Some were very advanced IT-wise, or others were, I've got an Excel spreadsheet of my phenotypes. And so we kind of had to make it. So that was one win that I think we did well. The other thing is when we first started, we had this notion that we want all of the data to be harmonized across studies, but we realized at the time that probably wasn't pragmatic. Well, possible. Let's say it that way. But it wasn't possible with the resources that we had and the curation staff we had. And the burden on the submitter would be too great to force that. So, and I've said this in other talks that I've given. The joke, and I think you'll appreciate this, is if you've seen one dbGaP study, you've seen one dbGaP study, because they're all different. And NCBI is part of the National Library of Medicine. And we kind of viewed it as each study is like a book. And we're not editors, but we are curators, and we can check that all the pages are there and that they add up. So one of the other, say probably the third good thing that we did had to do with developing a QA/QC of the incoming data that would not only check file format, things like that, and check things like if you called in one file this person a woman, and then in the pedigree file that person's a man, we know there's something wrong. Curators at dbGaP wouldn't know which way to go, so we'd have to go back to the submitters, but at least the scripts identified errors that we wouldn't have to answer with our help desk later. And that was a little bit of a departure from some of the other NCBI databases, which is kind of what goes in, goes out. And so within a study, we were very rigorous. Across studies, unfortunately, we decided early on, just because of the resource issues, that this might be feasible when we had critical mass data. And that, so I, really that combination of flexibility of submission, rigor within a study. And then I would say the third thing that we did pretty well is that we created dbGaP very modular, so that when sometime after, so dbGaP started right after HabMap. And so most of the data coming in were chip data,
Right.
which is, at the time seemed kind of large, but not until we saw the sequencing data. And that was a few years later, and we were adaptable to that. And really, we weren't going to recreate the sequence read archive. It was the general public sequence database for next-gen sequencing. Tell me when I get close to time. So we were adaptable, and because of our modular system, we were able to have a registration system where NIH officials would use it to register studies. There's a submission system that's completely separate, which people use to upload files to us. There's a request system for the PIs to use. There's public databases, and then that same request system actually has another interface that's separate for the data access committee to go on and approve or deny requests. And so, and then getting to kind of things that we didn't do well, and I'll acknowledge that, is I think really the data harmonization. We thought, we'll do this when we get critical. dbGaP now has about 3,000 studies, about 5 million study participants. Never really got to that. Kind of regret that we weren't able to harmonize across stage tests. Obviously, what that forces analysts to do is to harmonize every time, but if it could be done rigorously once, that'd be much better. I think, lessons learned are keep up on UI, UX, and technologies. And CBI didn't do that so well. We were kind of, and I don't want to go back to the resource issue, but we were kind of stuck into a certain technology, and that UI, UX kind of just went by the wayside on the data. And I kind of regret that. That hopefully will be fixed in the future. But I think overall, it really comes down to interoperability, change, and usability.
So that's what Mike is going to focus on doing now that he's at NCBI. No, NIDCR.
Yes.
My director was looking at it. Thank you. So as a data, as a clinician, a data intensive investigator for years, and now being a data scientist at NIDCR, also playing a lot of very active roles across the NIH to bring data intensive science forward. What has your previous experience, in different domains informed the actions you are taking now?
Yeah, thanks, Lou, for that question. I can speak as how it was as a trainee, where a lot of times you're just looking at resources available in the lab And I was in the School of Engineering, and there was not that much infrastructure in the DOC area and the dental oral craniofacial area for me to turn to. So I had to do a lot of my research on available resources, which was in other fields. So one of the first things that we did when I joined NIDCR was to take the data science working group's recommendations and build POC-specific infrastructure to find data and categorize it across different areas of informatics. There's a wealth of information in bioinformatics. Then when you get to EHR, there was a large gap there. Then once you get to imaging, it's just questionable the availability. So that's one of the main things that I'm taking from my experience trying to conduct research to creating resources that can help other folks in the same boat find the data, analyze the data, disseminate it. The second thing that I'll talk about is the lack of data quality and data quality metrics to evaluate the data that are available. Harmonization is one of the problems, and working with like rare diseases or cancer, the sample size becomes so small. If we try to find the right type of data, and there is often no documentation on the data, less on the analysis of the data. So this goes back to what we spoke about in the morning about the reproducibility of these pipelines. So there was this lack of documentation or guidance across the data lifecycle, right from generating it all the way to publication at the publication stage. So ensuring the availability of quality data and metadata is also a big priority for us because that was a big gap as a user. And now with the advent of AI modeling, that becomes a fundamental request from like almost all of the users that we meet is there adequate quality of data? And then also, is there a separate benchmarking data set where we can try out all of these models in a neutral ground? So that is also something that NIDCR hopes to prioritize soon.
Thank you. I'm going to give Vidhya a special treatment. Any quick question for Vidhya from the audience or from online? Not yet. We'll wait until the end of the session. So the last question goes to Carl. Axel, sorry. Didn't mean to ignore you. Axel has
I was off the hook.
No. My fault. So as the chair of our data science working group, increasingly and very productive, highly achieved data intensive investigator and our former council member, what specific advice do you have for the entire FaceBase program going forward?
Yeah, so I have a few thoughts. I'm not sure if all of them qualify as actual advice, but the first one, I guess, so the first thought that I have is to embrace data diversity. So this is something that became very clear through the work of the data science strategy working group. In the beginning, we tried to figure out what types of data to craniofacial dental researchers work with. And like we generated this list and it kept growing and growing longer and longer, right? So it was and probably still incomplete. So it does general data types, even more specific, like data standards and so on. And so thinking about a system like FaceBase, I think there's always a balance between building a well-structured system versus incorporating all of this diversity. And if you had asked me 10 years ago, I probably would have said, well, think very hard about what you're trying to incorporate, because there are these trade-offs, right? And you're at risk of just creating like a big mess of unconnected stuff in there, right? And however, looking at it today, I think my impression is looking at how data systems develop, that modern data systems to be good at bringing together different types of data, that they are also more forgiving in terms of, like, say, if there's data that has certain gaps, right? If there are metadata that don't perfectly align, that's still like bridging those gaps and bringing the data together. So I'm not sure how that translates into exact advice for anything that FaceBase is doing, but generally speaking, I think at this point, branching out to see if he can incorporate more data types that often come with new technologies. Now, if you think about, like, say, AI models that you want to, right, think about ways to incorporate that. The second thought is related to users, right? So database is only useful if we have users. And so I would just encourage the team to continue to do a lot of effort in education, outreach, getting the word out there. And again, this is not really advice because as we saw earlier, there's already a lot of workshops, there's great user support that is very responsive, but just essentially keep up the good job in that. Don't, de-emphasize that. I think, if anything, put even more effort into that. And then the first more specific one that came up when I talked to Justin, a frequent like a PI who has contributed a lot of data, right? And I think others have probably run into this issue too, is building awareness and recognition for FaceBase as a major and official repository for data. So within the community, I think everybody knows FaceBase, but so the specific situation that we discussed was when, like, a journal, had some new data and was in FaceBase where it's like properly, regulated with access and so on, but they just didn't know it, right? And they said, "Oh, can't you put it in dbGaP instead?"
Yeah.
Right. And I think it's just, again, it was eventually a result, but just a matter of building awareness, getting the word out there, maybe especially with major publishers. Maybe it's something that we'll just build over time, but keeping the back of mind. That's it for my end.
The advice we couldn't have. Even though I forgot momentarily. Thank you, Axel. I feel every panelist has been saying something that echoes with others so far. So that's, very encouraging. So now the final question goes to Carl. Being the FaceBase PI for years and being in your own computational space, from a lot of insightful lessons here today, I have a specific question for you. What should FaceBase be doing to multiply the value of data that it manages and shares? Let's say, with outcome we'll all see.
So actually, that's something I think we've been thinking about a lot. And I think it kind of intersects with some of the future directions we've been talking about. I think that multiplication will happen at two levels. So one is we keep on building a robust, highly curated, respectable corpus of data and expand that. Along with, I think it was Axel's comments, I think we need to walk very carefully. One of our great successes is we've been focused and not boil the ocean in terms of what we do. I mean, Mike, to your comment, right? That's allowed us to do some practical, pragmatic integration of the data across modalities. So I think we should, expand the corpus, but kind of walk that line so we're not something to everyone. We maintain kind of a core focus of activity, so we don't become too slow, too bureaucratic, too inflexible. Then I think then the other thing is, so we kind of build this foundation. Then I think the other thing is we need to be looking at the full life cycle and increasing the number of loops. So I think that we need to be looking at the foundational data, and then I think we can play a real role in intermediate and derived data products, which I think are going to become increasingly important with all this machine learning stuff, right? So the idea is not to take one piece of data, produce one result, and then the answer is 3, right? But I think we'll see emerging derived data sets. This goes to the benchmarking as well. That will be seeing curated derived data sets that will be the result of these other computational methods and feeding in. So I think that's a multiplying factor. I think another multiplier, which was mentioned here, that we're trying very hard to look at is kind of the usability. I think we've got a really sound foundation. We've always been really concerned, and I think one of the things we've been pretty successful on is we always talk about FaceBase as a social technical system. So we've always been very interested of human in the loop right from day one. We've done these usability studies and so on. So I think keep on hammering on that really hard. And then as we discussed also the education, I think now especially with all these new methods and methodology coming on, what can we do to train people so that they are consuming and producing data that's experimentally valid and scientifically rigorous? I think we dropped the ball a little bit. That I think there's a lot of computational results coming out in the past by people who, I think dumbing down tools ends up with dumb results. We need to figure out education, smart and usable tools so that we produce high quality results, right? And then work on building this culture, which we've made some progress. Data citation, value in data, use of data in tenure promotion cases, getting funding to support data validation and reproducibility experiments, kind of things we've been talking about for a while. But I think all those things can also go into multiplier effects. Right? And then maybe my last thought is, as I'm just rambling here, think of this system-wide, right? That we don't pick one thing. Look at the system, all the points of control we have. Let's pick up the ones that have the biggest impact that we can change. Let's work on those, and then keep on continuously evaluating, reassessing, continuous improvement, right? That's what we did in engineering way back, right? It's continuous process improvement, right? And that's the way I think we'll build these multiplier effects. I should just maybe just rambling last comment, when multiplier effects, when we went from, was it phase 2 to phase 3, right? We went from the hub and spoke, Rob mentioned this, hub and spoke model to the network model, right? I was scared to death that we would fail miserably, right? Because we had 10, and I don't know if people remember the early days, even though we paid people to give us data, they wouldn't give us the data, right? That there were incidents where people would not, under threat of funding termination, they didn't provide the data. And I won't mention any names, but Yang knows who I'm talking about. Right, so I was really scared multiplier. What's that? Present company excluded, wasn't Yang. But so one of my big kind of satisfactions over the last several years is that we didn't fall into a cliff, that we built this kind of flywheel and this increased visibility and contribution, and that what I thought was going to be a huge problem is not, right?
And the community becomes the oil.
Yeah, exactly. So, I would say to your question about multiplier effect, I think we were very deliberate about those mechanisms. I think a lot of the things we did have turned out to work well. I think we need to continue to refine and look for new mechanisms for doing that. But one can only be optimistic that we didn't fall into a big pit and have 12 data sets, 5 years later. And I was really worried about that, and that turned out to be the least of our problems. So
thank you, Carl. Yeah, that was fascinating.
Yeah.
How much time do we have left? Give us a few minutes, please.
5 minutes.
5 minutes. 5 minutes. And to our audience in the room and online, what else should FaceBase be doing to multiply the FaceBase data's value?
And can I add on a question to that? And it's open to the panelists too. As we grow this data types, representation, and multimodality, and I was thinking about you, Axel, what is the role of a repository versus something like DDS Hub, which is a knowledge base? How can these two complement each other? So the repository is not doing all of the heavy lift of a multimodal representation, and it can be supplemented by a knowledge base or other. And Anup, if you have thoughts on this, I would love to hear.
Maybe you go first because I don't have a good. Well, I'm not sure how to do it best. I mean, the way I think of this is so like as you get in more different types of data that are inherently unconnected, you rely on the ability of future systems to be able to make those connections, right? If we think about, say, agentic AI, where you expose the data and then it can make those connections. For that, it is of course very important to design the systems in a way that they are AI ready, whatever that means.
Because sometimes
from my understanding, mostly following things that should have been good practice before, which is common standards, properly documented metadata.
And in the lack of those standards, when you are not able to harmonize. And that goes back to your comment. It's like, do you restrict what data you accept? And I think the answer you're saying is no, but reverse engineer it.
Yeah.
I think these systems.
But we heard also from Carl, though, that one, like at least in the past, that's what I meant when I said like 10 years ago, I would have said something different. And maybe to some extent still today, like the one aspect of success is in being somewhat selective in what to accept. So what I don't know is where is the right balance. I think, what I try to say with my comment is that I think over time, it will move in a direction that you're increasingly able to accept like different types of modalities because the systems will be able to accommodate that in a more independent way. But it will, yes, there will always be some trade-offs.
Not to that, the other thing to keep in mind is that the time between data generation and knowledge generation can actually be years in between, right? So if a data repository is trying to build knowledge, the inherent nature of it is not possible. I'll give an example of the BRAIN initiative, right? So we run a data repository for BRAIN initiative where the data was generated 5 years ago and the actual cell typing happened 3 or 4 years after the data was deposited. So if you were to try to expect the knowledge base to know all that information, it's not possible. So by that nature, keeping those two separate actually helps.
And also, isn't it more or less true that for knowledge base, you have, published discoveries or knowledge sources acceptable to you to be synthesized?
Yes.
There probably is less of an expectation for knowledge base to be able to anticipate what's coming to either harmonize or normalize. But as a data repository, if the data types do not talk, then what's the usability? Sort of to me means different requirement of the expertise. Pedro?
Just had a very basic question. As we're looking at AI-ready data and these chatbots and things, is there a reproducibility index or like a way the bot can say, that's a good question, but there's no data on this versus you have one data point and here's your graph.
Is there a way or should there be a way? What's your question?
I think sort of Axel and as we think of the next wave of using this data, I want to get people ready to use it, but I'd love to get them some feedback to say, we need 10 more data sets or 1,000 more data sets to answer your question.
Doesn't that question also rely on some index on the reproducibility?
So one thing to sort of respond to is transparency, right? So one idea would be if somebody runs a question, we can provide what is the backup information from which that information is derived. That would sort of create the transparency that's needed. So if it's coming from one experiment with one replicate, right, the quality is going to be different than, let's say, a big atlas that was created with hundreds of replicates. So there's, I feel like that transparency should be up front and center when we provide information. That's one way of addressing it.
I would say that generally, as AI methods become more prevalent, trust is a major thing that people think about, right? Where, whenever you have used any of these AI-based tools and you get results, can you trust it, right? And there are two components to this. One is a social, and the other one is or psychological component, which is humans trusting AI in general, which is the same consideration that applies to like anyone believing what their ChatGPT thing tells them about whatever problem. But the other one is one that can actually be measured, right? Which is technical trust, where you essentially take the responses and then compare it with objectively verifiable methods to see how well does it align. And from that, you can think about like deriving things such as an index or some kind of measure of that trustworthiness. I'm not sure that anyone has come up with a generalized approach for doing this.
Well, I mean, this is
Another minute.
I mean, this is a question I've been looking at a lot, and I think it's not an AI question, machine learning question. I think it's a science question, right? And I go back to I've been spending a lot of time looking at the discovery of modern physics and particle physics, and what it meant to do an experiment and say, okay, the charge on an electron is X, right? And the documentation, the reproducibility, and what went behind that, right? So I think we need to, as a community, understand, running these models, that's an experimental regime. What does it mean to have a hypothesis and generate a testable, reproducible result on that hypothesis? And rather than using a particle accelerator, or rather than using a whatever, I have to be using computer as my experimental apparatus. So I think we need to kind of philosophically, we're doing science, we know what it means to do science, we need to apply that same rigor and methodology and figure out how to adopt that to the tools or the instruments we have today for conducting these experiments. So that's something we're looking at very deeply.
All right, to be continued. Thanks to our panelists.